Welcome back to a familiar course in an unfamiliar setting. We're going to take Chapter 21 right from the top since it's been a while. Please remember to post questions to the forum on Moodle as you have them, and I'll answer them for everyone there. As a reminder, these intro slides are a good high-level overview of what we are learning, so you can always refer back to them. Also remember that you have reading guides and lecture notes posted in Moodle, and you'll get the most out of these videos if you've done the reading as assigned. We're talking about enzymes in Chapter 21, and remember that enzymes are a form that proteins take, one out of many forms that the proteins take. And because vitamins are often associated with enzyme functions, we'll talk about vitamins in this section as well. Enzymes are any organic compounds that act as catalysts for a reaction, and most of the time they're proteins. We'll talk about a few special examples in Chapter 22 where ribosomes function as enzymes. Some of the nomenclature that we had talked about in proteins in Chapter 20 is a little bit different here in Chapter 21. So in Chapter 20, when we talked about protein classification, we might have talked about monomeric or multimeric proteins, monomeric being just one polypeptide chain and multimeric being multiple peptide chains. And then we talked about simple versus conjugated, simple being only amino acids and conjugated having something other than amino acids, a prosthetic group. We use those same terms to refer to enzymes, but we also get a little bit more specific when we talk about the prosthetic groups. The prosthetic groups for enzymes are called cofactors when we're talking about enzymes. So a cofactor is a specific example of a prosthetic group. When we talk about an enzyme then that has the cofactor, the enzyme with the cofactor is called the holoenzyme, think like whole enzyme, and the enzyme without the cofactor is the apoenzyme. We can also further narrow down cofactors so that cofactors can be designated as inorganic or organic. The cofactors that are inorganic we still will call a cofactor. Some really common examples are like magnesium, which is responsible as a cofactor for many enzymes that deal with DNA. If cofactors are organic molecules, we call it a coenzyme, and most vitamins are coenzymes, and so this is something that we'll spend some time talking about at the end of this chapter. When we name enzymes, their names will either end in "ase" or in "in". Most commonly, enzyme names end in "ase". That refers to the fact that they have catalytic activity. Some enzymes will end in "in", especially if they were discovered long ago. So if you see a word that ends in "ase" or "in", you should know that it's an enzyme. The enzyme name might be made up of one or two words. If the enzyme name is made up of two words, the first word always indicates the substrate. That's what the enzyme is acting on. The second word will always indicate what type of reaction is happening on that substrate. If the enzyme is made up of only one word for its name, the reaction is always hydrolysis, and the substrate is just going to be the prefix. So for example, we should be able to identify what each of the following enzymes is doing, at least in general. So the first enzyme, cellobiase, is just one word, so we know that it's going to hydrolyze, and it's going to hydrolyze something that's beginning sounds something like cellobi. We know the molecule cellobiose. Cellobiase hydrolyzes cellobiose. This works whether or not the enzyme is actually something you know, or if it's something completely unique. So for example, textbook disintegrase, the reaction would be a disintegration. It might disintegrate your textbook. We don't actually need to know anything about the enzyme to know what its function is. We only need to know its name. While many of the reactions that we need to identify are reactions that we talked about all the way back in organic chemistry, it's helpful to have a reminder of the types of scopes that enzymes can have and that you'll see in their names. So for example, we have 6 major classes of enzyme nomenclature, each of those having subclasses that refer to specific reactivity as well. The first class is oxidoreductases. They're responsible for oxidation and reduction. Transferases are responsible for moving a group from one place to another. Hydrolases are responsible for adding water to break things into pieces. Lyases are oftentimes responsible for forming double bonds, either by removal of water or removal of carbon dioxide. Isomerases will change a molecule from one isomer to another isomer, and ligases are going to join two pieces together to make a new, larger molecule. Almost always these require energy, and almost always these require ATP. We absolutely need to know the 6 major classes of enzymes, but within those classes, it's also important to know certain subcategories that occur very often biochemically. So again, the first class of compounds, of enzymes that we need to recognize are oxidoreductases. Oxidoreductases perform redox reactions, which we talked about in chapter 9.3 of Gen Chem, but we also talked about it in chapter 14. Primary and secondary alcohols can be oxidized. We talked about it in chapter 15. Aldehydes can be oxidized, ketones cannot. And we talked about in chapter 16 as a way that carboxylic acids are produced. Generally, we see increases in the number of bonds to oxygen or decreases in the number of bonds to oxygen will indicate an oxidation or a reduction. We also see that oxidations and reductions occur when we move between an alkane, a single bond between carbons, and an alkene. Transferases are a second class where we move a functional group from one molecule to another. Two types of transferases that occur very often in biology are transaminases, which are responsible for moving amines and processing amino acids in our amino acid metabolism. Again, transaminases are really important to remember because of their central role in amino acid metabolism. And kinases are really important because they move phosphate groups. Now, a kinase is a transferase that moves phosphate groups, which means the phosphate group is moving from one molecule to another molecule. That molecule might be ATP, or it might be any other molecule. Hydrolases are performing hydrolysis, where we add water to break a large molecule down into smaller molecules. That can happen with our 3 major biological molecules: carbohydrates, lipids, and proteins or peptides. When we add water to break down a carbohydrate, it will break the acetal or the glycoside bond, and we call those enzymes carbohydrases. When we add water to a lipid, that will break an ester bond for our saponifiable lipids, and we call those types of enzymes that perform that reaction lipases. And when we add water to a protein or peptide, it will break the amide bond that forms the primary structure of that protein or peptide, and we call those proteases. We also have lyases. Lyases are perform addition or elimination reactions. Think of the reactions that we talked about with chapter 13 as addition reactions. They are either adding on to a double bond or they're eliminating to form a double bond. Usually we're talking about adding on water or adding on an amine, or eliminating water or an amine. We also sometimes will eliminate carbon dioxide in lyases to form an amine. Isomerases are going to perform reactions that yield an isomer of the original product, so it's going to have the same formula, but it will have a different structure. These are fairly common in our central metabolism. And ligases are going to perform reactions that combine 2 molecules in one. Notice that ligases require ATP as an energy source, but the phosphate is not transferred onto any of the molecules. Only the energy to break ATP apart is used to combine those molecules into one. Shown on the screen are several examples of reactions that you should be able to look at and identify what type of reaction is occurring, which major class of reactions is at play, or which major class of enzymes is at play. So if we look at the first reaction, we can see that we have an alcohol at this location that is becoming a ketone at that same location in the product. Because an alcohol only has a single bond to oxygen, and now that same location has a double bond to oxygen, we know that we have an oxidoreductase as the enzyme at work here. In the second reaction, we see that we're adding water to a molecule to break it into 2 smaller pieces. What are those pieces? Well, each of those pieces has an amine, and then we have a carbon, and we have a carboxylic acid. So each of those pieces is an amino acid. We are taking a group of amino acids that are linked together and making 2 individual amino acids. This type of reaction is catalyzed by a hydrolase, adding water to break things into pieces, specifically a protease, where we take a protein, a polymer of amino acids, and break it down into its amino acids. The next reaction has a fructose 6-phosphate, which becomes a fructose 1,6-bisphosphate. So we're moving a phosphate onto the molecule. You can see on the fructose 6-phosphate, this alcohol is free, and on the fructose 1,6-bisphosphate, we've now hooked a phosphate onto that alcohol position. That phosphate came from ATP. That doesn't mean that we're spending the ATP just to join the molecules together. We're actually moving the phosphate onto the molecule, which is why this is classified as a transferase as opposed to a ligase. The final molecule is going to eliminate water and form a double bond. So in this final reaction, we have the elimination of water and the formation of a double bond. When we have a double bond formed by eliminating water, or when we add water to a double bond, we should think of a lyase always being the type of enzyme at work. We have several models of enzyme activity that can help us to understand what's going on in the enzyme itself. The first of those is the lock and key model, which we can see in the top row. We see that substrate can bind to an active site that's perfectly formed to fit that substrate. Once it's bound, it forms what we call an enzyme substrate complex, and that enzyme substrate complex can catalyze the conversion of the substrate into a product or multiple products. We also can talk about a model where the enzyme active site doesn't exactly fit the substrate, but it's pretty close. Once the enzyme and substrate bind together and we have the enzyme substrate complex, the enzyme substrate complex has some amount of strain that helps to catalyze a conversion to products. That's called the induced fit. Some models apply better to some enzymes and others to other enzymes, but in reality the truth is somewhere in between. We have not exactly a perfect fit, but it's very close in most cases. The thing that is consistent between both of these models is that both models require an active site. That's the area of an enzyme where the substrate binds and the reaction takes place. If we do not have an active site, we cannot have any sort of enzyme activity. The reaction rate will be almost nothing. And so all of the types of things that unfold proteins will also unfold enzymes, since enzymes tend to be proteins. And when those proteins unfold or are denatured, when the enzymes are denatured, we see that there is no active site and no enzyme activity. Enzyme active sites will be formed by the three-dimensional structure of the enzyme protein itself. The three-dimensional structure will have an active site that has R group interactions or side chain interactions that are tailored to match the substrate. By tailoring these interactions to match the substrate to different levels, the enzyme is able to deliver different amounts of specificity. Some enzymes will be absolutely specific or absolutely selective. Absolutely selective enzymes will react on exactly one substrate only. While this is relatively rare, it's actually really important for our central metabolism because we want our central metabolism to perform specifically one reaction without very many side reactions. We also might have stereochemical selectivity. In stereochemical selectivity, we will only react one set of stereoisomers, but not the other. So for example, we might have an enzyme that can react on all carbohydrates that are D, but no carbohydrates that are L. This allows that enzyme to have a wide-ranging scope of action, but it will only react on the naturally occurring carbohydrates. An example of this is alpha amylase, which is the enzyme in our spit and in our intestines that breaks down polysaccharides. It can react on any length of starch, but it will only react on the D carbohydrates, on D amylose. We can also see group specificity. Group specificity is where we only react on one set of functional groups, so like amino acids, or linkage specificity where we only have one type of bond. So we might have acetyl functional groups that are not glycoside bonds that can't be reacted by an enzyme with linkage specificity, but they might react with glycoside bonds specifically. Enzymes not only have selectivity for different types of reagents, they also have varying levels of activity depending on what type of environment that enzyme is in. There are four major factors that affect the rate of enzyme activity, the first of which is temperature, which we can see displayed on the left. We notice that as temperature increases, initially we're going to increase the reaction rate in this part of the graph. But once we reach an optimum temperature, then we switch and we end up having a decrease in enzyme reaction rate. When we're increasing the reaction rate, what's shown in yellow, that's usually because we're increasing the overall kinetic energy. Remember that for a reaction to occur, it requires us to have a certain amount of energy, and the higher the fraction of molecules that have sufficient energy, the faster the reaction occurs. So when temperature increases, more reactions occur because there are more molecules with sufficient energy. However, as temperature continues to increase, we meet a point where enzymes begin to denature, they begin to unfold. And when enzymes unfold, they no longer have an active site. Without an active site, they can no longer catalyze the reaction, and so we see denaturation will start to cause enzyme rates to go down because the enzymes are no longer active, what's shown in red. The optimum temperature can be different depending on which enzyme we're talking about. Most often, those enzyme optimum temperatures match the environment the enzyme is likely to be in. So for humans, most of our enzymes are optimized for 37 degrees Celsius or 98.6 Fahrenheit because that is the environment that our enzymes work in. We're going to do an enzyme lab, which we won't do anymore, where we have potatoes. The potatoes enzymes are optimized to be most active at temperatures that potatoes are likely to encounter, like around maybe 30 degrees Fahrenheit to 70 degrees Fahrenheit soil temperature. We also see that pH will affect the enzyme rate. pH will affect the enzyme rate going down from an optimum pH on either side. So if we have an enzyme that is below the optimum pH, the increase in acidity, lower pHs, higher acidity, the increase in acidity will protonate more of the side chains and cause the protein to unfold or denature. The same thing happens if we decrease the acidity or increase the pH. The protein begins to unfold, denature, and we no longer have an active site. So there's some optimum pH where the protein remains folded, the enzyme remains folded, and we have a maximum rate. Again, this maximum rate is going to match the type of environment that the enzyme is likely to be in. So the maximum rate pH is going to be around 1 for our stomachs that have a pH of around 1, but since our bloodstream is around 7.4 for a pH, most of the enzymes in our bloodstream are optimized for a pH of 7.4. We also can see a change in rate as we increase the amount of substrate concentration, which is the next graph over. Notice that as substrate concentration increases, at the beginning, we see the reaction rate go up very drastically. But as we continue to increase substrate more and more, the reaction rate levels out until it approaches some maximum reaction rate. We can never go faster than the maximum reaction rate. This is the maximum theoretical efficiency for that enzyme. We can think of this kind of like a factory worker. A factory worker can only work at some amount of maximum rate. If the factory worker has all the supplies the worker needs, the worker can work close to that maximum rate. If the factory worker does not have all the supplies it needs, if the substrate is low, then the reaction rate will be significantly less. And as substrate becomes available, as materials for the factory worker become available, the reaction rate quickly increases towards that maximum rate. And of course, we can also see that reaction rate will increase as we increase the enzyme concentration. We can use the same factory worker example to talk about this. If we have more enzyme, it's like having more factory workers. And the more workers that you have, the faster they can work. We can also affect rate by using enzyme inhibitors. Enzyme inhibitors are molecules that are going to decrease the normal level of activity by binding to it. Oftentimes, we will introduce enzyme inhibitors into a system. It is very rare for organisms to produce their own inhibitors. Most drugs are enzyme inhibitors. There are three different forms that inhibitors tend to take. They're either competitive, non-competitive, or irreversible inhibitors. Competitive inhibitors are when we take something that looks very similar to the substrate and we bind it to the active site instead of the substrate. As long as the competitive inhibitor is in the active site, the substrate can't bind. Because the competitive inhibitors bind reversibly, the higher the competitive inhibitor's concentration goes, the more the substrate will be excluded. An example of a competitive enzyme inhibitor is using ethanol to prevent methanol from being oxidized in alcohol dehydrogenase. When methanol is oxidized in alcohol dehydrogenase, an enzyme, it becomes formaldehyde and it can fix our cells. It's very toxic. If we use ethanol in high concentrations, it prevents the methanol from being oxidized. The ethanol gets oxidized instead, and we get really drunk, but it keeps us from going blind. Non-competitive enzyme inhibitors work a little bit differently. Instead of binding at the active site, the non-competitive inhibitors bind at a different site on the enzyme. When they bind, it changes the activity of the enzyme. It sometimes locks in a substrate, it sometimes locks out a substrate, but the enzyme can no longer function the same way it did before. And because it's bound at another site, the substrate can still bind, but it won't react in the same way. The active site becomes less effective. Finally, we have irreversible enzyme inhibitors. Like competitive enzyme inhibitors, they bind at the active site, but when they bind, they're actually covalently linked. So that link is there forever. Once we have an irreversible inhibitor bound, that enzyme can no longer function, no matter what. The enzyme has to be broken down to amino acids and resynthesized in order for the system to regain any sort of activity. These inhibitors then can be very powerful because the enzyme is completely deactivated. While we introduce artificial regulation of enzyme activity with inhibitors, our cells need to be able to do this naturally, because sometimes there's not enough substrate for our cells to work on or for specific enzymes to work on. Our cells need to be able to turn off that enzyme when there's not enough substrate and turn it back on when the substrate comes back. Or if there's too much product, for example, if we have too much ATP, our cell needs to be able to tell its enzymes to slow down and use whatever we have to make something else instead of ATP. We can do this within our cells in three different ways. The first is allosteric enzymes that are under feedback control. In a very broad sense, this is when we have a molecule that either turns on or off enzymes. We'll talk more about this in the next slide. Enzymes also can be activated by other enzymes. This is really helpful when our enzymes do something that we don't want them to do. We need to turn them off initially and turn them on later when they're in the right environment. Finally, we modify our enzymes covalently. We add another group on, a prosthetic group, oftentimes a phosphate that's added using ATP or an oligosaccharide. In each of those cases, the phosphate or oligosaccharide are really polar and can change the overall structure of the enzyme, either turning it on or turning it off. Allosteric enzymes, again, enzymes that are able to be regulated by other molecules, have a couple of interesting characteristics. They almost always have quaternary structure. They have separate binding sites for substrate and regulator. So notice there's a binding site and active site, and there's also a site where the regulator can bind on. Notice the regulator might be negative or positive. So when a regulator binds on, it will change the protein structure, and it might make it so the active site is no longer there, make it slower, or it might make it so the active site is actually better. Both can be true with allosteric enzymes, which is different than inhibitors. Inhibitors will only slow down the rate of an enzyme. Allosteric regulators can either be negative and slow down the rate of an enzyme, or they can be positive and speed up the rate of an enzyme. Feedback control is a specific mechanism where we use a product of a reaction to slow down an earlier enzyme. This can be really helpful in a metabolic sequence. So for example, if we're taking glucose and eventually making ATP, we might not want to use up all our glucose if we have a lot of ATP. So think of this product D like ATP. If we have lots of ATP, that ATP can come back and bind on to enzyme 1, telling enzyme 1 that it needs to slow down. The enzyme 1 then will be able to slow down as it waits for the ATP to be used up for other reactions, keeping the glucose for later. Our second example of ways in which our cells regulate protein activity or enzyme activity is by using proteolytic enzymes. Proteolytic enzymes are enzymes that break peptide bonds. Usually those are really dangerous to have produced in our bodies in an active form, because the same place that those proteins are produced are where all the other proteins are produced. And if they break peptide bonds, break proteins apart, we would produce all sorts of proteins and break them apart all at once. Proteolytic enzymes then are produced inactively. They're moved into the lysozyme where all proteins are broken down, and once they're in the lysozyme, they'll be converted to an active form by breaking out a protein fragment. So this zymogen, or proenzyme, the inactive form of the proteolytic enzyme, has this peptide fragment that keeps it from actually functioning. As soon as it enters the lysozyme, that piece can be cut out, and now the protein is going to fold the right way to start cleaving other proteins. Covalent modification is the last way where our bodies and cells are able to regulate enzyme activity. This is accomplished by attaching some sort of prosthetic group onto the enzyme. Usually that attachment is reversible, although it's covalent, so it's relatively permanent. The most common form of covalent modification is phosphorylation. Phosphorylation will take phosphate from an ATP and move it onto the enzyme. Another enzyme will often catalyze this. Those enzymes are called kinases because kinases are the transferases that move phosphate from one molecule to another, in this case from ATP onto an enzyme. Again, covalent modification can change the rate of an enzyme either up or down. It depends on the enzyme. We've now talked about artificial ways that we inhibit enzyme activity and also natural ways that we regulate enzyme activity. In the next few slides, we'll talk about examples where we will inhibit enzyme activity with drugs, and we'll talk about specific drugs and how they work. The first example we're going to talk about is an ACE inhibitor. ACE is really just short for angiotensin converting enzyme. That's an enzyme that's going to convert an inactive hormone, angiotensinogen, into the active form of the hormone, angiotensin. The active form of that hormone constricts blood vessels and elevates blood pressure. So, if we are inhibiting ACE, which makes angiotensin, what effect do you think the ACE inhibitor has on blood pressure? Pause for a moment and see if you can answer that. The ACE inhibitor will prevent angiotensinogen from becoming angiotensin. And since the ACE inhibitor prevents angiotensin from forming, all the effects of angiotensin are blocked. Since angiotensin elevates blood pressure, an ACE inhibitor has the net effect of decreasing blood pressure. Another really important set of examples of inhibitors are antibiotics. The first class of antibiotics that we're going to talk about are the sulfa drugs. Antibiotics are really any molecule that kill or weaken bacteria. Oftentimes we accomplish this by targeting an enzyme that the bacteria need, but humans do not. Sulfa drugs are going to be competitive inhibitors for the bacterial production of folic acid. Folic acid is produced by using para-aminobenzoic acid, whose structure you can see over on the left. Since all the sulfa drugs have a very similar structure, they can bind into the same active site and prevent the enzyme from functioning on the para-aminobenzoic acid. Since folic acid is required for bacteria, but we get it from our diets, it doesn't affect us at all. Another class of antibiotics are the beta-lactams. Beta-lactams are molecules that are based on penicillin, and they all have the same core structure where there is a beta-lactam. That beta-lactam fits perfectly into a transpeptidase enzyme that links together and strengthens cell walls in certain types of bacteria. Without that function, bacteria are far more vulnerable to our native immune function. However, it doesn't kill off the bacteria, and so the bacteria are readily able to develop resistance in many cases. Beta-lactams are really specific to the active site, and the bacteria are easily able to alter that active site to make that enzyme resistant to the drug. Some bacteria also produce a protein that hydrolyzes the amide of beta-lactams before they interact with the transpeptidase, which makes them overall useless. They call that enzyme a beta-lactamase, meaning that beta-lactams are the substrate and beta-lactamase would be hydrolyzing the beta-lactams. Since my daughter has been having plenty of ear infections, I've been pretty familiar with the beta-lactam family of medicines, but also with clavulanic acid, which is a beta-lactamase inhibitor. Remember, beta-lactamases are the enzymes that degrade the beta-lactam antibiotics. Clavulanic acid is an inhibitor of those enzymes. So clavulanic acid doesn't have any antibiotic activity of its own, even though it has a beta-lactam, but it's used along with amoxicillin to stave off resistance by preventing the beta-lactamase activity. Together, those are marketed as augmentin. Another antibiotic is Cipro. Cipro attacks DNA gyrase, which is an enzyme that's present in bacteria but not in humans. Its mechanism is unknown. DNA gyrase is essential for bacteria to have their DNA in the correct form to replicate, which means it doesn't actually kill off the bacteria, but it keeps them from copying at all. Since the bacteria can't copy, they can't develop a resistance, and so they don't develop a resistance very easily at all. And Cipro is really widely active against a lot of bacteria, a quality that we refer to as broad spectrum. It's kind of a last line of defense in many cases. Another really important enzyme in overall drug metabolism is the class of enzymes called cytochrome P450s. It's not just one enzyme, it's actually a huge group of enzymes that are for degrading all sorts of compounds that we can't metabolize under other pathways. Drugs fall into this category as well as many other molecules. Grapefruit actually contains natural inhibitors of cytochrome P450. So if you have grapefruit, the cytochrome P50s aren't able to degrade the drugs, and then the drug stays in your system much longer, resulting in much higher doses than are expected. Not only are enzymes targets of inhibitors to treat diseases, enzymes themselves can be either indicators of diseases or they can be used to treat diseases. So for example, an abnormal enzyme level in the bloodstream can oftentimes indicate damage to a certain type of tissue. So enzymes that are present in tissues that are then released into the bloodstream would indicate damage to that tissue. So enzymes like lactate dehydrogenase, which are present in the heart or liver, showing up in the blood, indicate huge problems in either our heart or our liver. Enzymes can also be used to treat diseases. For example, people who are lactose intolerant lack lactase in order to degrade the lactose. If lactase is introduced into the diet or is used to treat the foods before ingestion, the lactose is degraded earlier and people can then have dairy products. Enzymes also can be used to convert molecules into one form or another, and signals from that conversion or the resulting converted molecule can be much easier to detect. A really common example of that is a blood glucose meter. It's hard to detect glucose, but glucose dehydrogenase will change the glucose into a different form, and that change registers on the blood glucose meter. We've now talked a lot about enzymes, but many of our enzymes require cofactors in order to properly function. Those cofactors that are small organic molecules that are not able to be produced by our bodies naturally are called vitamins. Vitamins are required in really small concentrations, especially compared to our proteins, carbohydrates, and lipids. So while they are required in our diets, we need way less of them, about 6 orders of magnitude less from them. So a microgram scale as opposed to a gram scale. There are 13 vitamins. 4 of those vitamins are fat soluble, meaning they're largely nonpolar, and 9 of them are water soluble. The vitamins that are fat soluble tend to stick around for a lot longer, and we need them in periodic doses. The ones that are water soluble tend to get eliminated much faster with our urine, and because of that, we need them in much more frequent doses. The first of our water-soluble vitamins is vitamin C. Vitamin C is also called ascorbic acid, so many times on an ingredients list you'll see ascorbic acid. In most species, ascorbic acid or vitamin C is synthesized naturally from the carbohydrate glucose. However, in humans, it is not possible to synthesize vitamin C from glucose, and so it's essential. Vitamin C plays an important role in reducing iron to help maintain proper collagen structure. Remember, collagen is the fibrous structural protein that allows our skin and many other tissues to hold together. Vitamin C also is a general antioxidant, and because of that, it has a broad-ranging function. It's also helpful for maintaining the proper form of other vitamins like vitamin E, which also functions as an antioxidant. A second set of water-soluble vitamins are the B vitamins. There are actually 8 B vitamins that are numbered 1 through 12. And the B vitamins are not retained with the proteins, so they're generally required in really small concentrations. Proper level of these vitamins are essential for proper function. Each one of the B vitamins actually has a different role in the body. We're not going to get more specific than that. One thing that's interesting about the B vitamins is the B vitamins are oftentimes supplied through a wide range of protein-rich foods. Generally, the types of things that are most rich in protein are the types of things that are good sources of B vitamins. Many times vegetable sources are good sources for B vitamins, with the exception of vitamin B12. Notice that vitamin B12 has no significant vegetable sources, and so people on a vegan or a vegetarian diet need to worry most about B12 levels. And oftentimes we'll take B12 supplements. In addition to vitamin C and the 8 B vitamins, there are also 4 fat-soluble vitamins, the first of which is vitamin A. Vitamin A actually comes in several forms. All of those forms are responsible for roles such as seen in color, the differentiation of cells, the development of epithelial tissue, and reproduction. The A vitamins are actually oftentimes derived from carotenes, like beta-carotene, which is in carrots. It makes carrots orange. A second set of fat-soluble vitamins are the D vitamins. D vitamins are the one type of vitamin that actually can be synthesized in our bodies. They're synthesized from steroids with UV light. So we can synthesize vitamin D by going out into the sunlight. People who have less pigment in their skin usually are more susceptible to damage from UV light, but are also better able to synthesize vitamin D from those steroids. People who have more pigment in their skin typically have more problems synthesizing D vitamins. However, most of us don't get enough sunlight to properly supply the D vitamins that we need for our maintaining proper levels of calcium and phosphate. And so oftentimes D vitamins are supplied through dairy, where we often would get our calcium and phosphate, and they are also supplied artificially as vitamins. Most people are D vitamin deficient. The last 2 fat-soluble vitamins are the E vitamins and the K vitamins. The E vitamins function as antioxidants, much like vitamin C. However, the E vitamins are much less powerful as antioxidants than vitamin C, so vitamin C can actually take the oxidized form of the E vitamin and reduce it back, and then the E vitamin can turn around and reduce other things. That allows us to have much lower levels of E vitamins if we have a higher level of vitamin C. K vitamins are responsible for proper folding of many proteins, especially the clot-forming proteins. So K vitamins oftentimes are help to regulate clotting.